Good afternoon, everyone. Today I am going to tell about immunology from BTR. So I will explain important points in immunology. Okay, so let's start with inflammation first. What are the sequence of events in inflammation? First, there will be transient vasoconstriction. Actually, in inflammation, main problem is vasodilation, but here first there is small amount of vasoconstriction which is within seconds. Okay, it is a neurogenic reflex and it is endothelium mediated reflex. Okay, so what is transient vasoconstriction? It is a neurogenic reflex which is an endothelium mediated. Okay, and when the blood vessels vasodilate, there is release of several inflammatory mediators like histamine, nitric oxide, prostaglandins, which increases the blood flow and cause redness, rubor, and heat, calor. Okay. Afterwards, there is increase in vascular permeability, which occurs mainly in the post capillary venules, which is extravasation. So what is the main mechanism for increasing in vascular permeability? It is endothelial retraction and endothelial injury. What is endothelial retraction? Here it is mediated by histamine and bradykinin, and this effect of endothelial retraction means endothelial cells will come far. So this is mediated by histamine, bradykinin. So this will last for minutes. And what is endothelial injury? It is seen in burns or toxin, and it will last hours or 2 days, which is a sustained effect. Okay, so repeat with me again. So what are the sequence of events in inflammation? First, in inflammation, mainly vasodilation happens. There will be release of mediators due to increasing vascular permeability. But before the vasodilation, there is within seconds, some amount of vasoconstriction happening, which is a neurogenic reflex and which is endothelium mediated. Okay, when vasodilation happens, there are a lot of mediators released like histamine, nitric oxide, prostaglandins. Okay, and this due to the increased blood flow, you will see rubor, calor, all that. Okay, afterwards, this increasing in vascular permeability happens where in the post capillary venules there will be increase in vascular permeability. Why this increase in vascular permeability happens? There is endothelial retraction, means the endothelial cells will come a little far, which is a transient effect, and this endothelial retraction is mediated by histamine and bradykinin. Next, in endothelial injury, what happens? This is a case which happens in burns and toxin, and which is a sustained effect which will last for hours and days. Okay, so now you got all the sequences of events in inflammation. First is transient vasoconstriction, next vasodilation, and increased vascular permeability due to endothelial retraction, and also due to endothelial injury, which is happening in burns and which is a sustained effect. Okay, now coming to chemokines. Okay, what are the chemokines? Classification will learn. Okay, first is alpha, which is CXC, which is mediated by interleukin 8. CXC, X is like 8, so X is if you write X, it looks like 8, so it is mediated by it is just as mnemonic to remember CXC. Okay, CXC is like interleukin 8, it is mediated by neutrophils, and CC beta is eotaxin. Okay, I told you what is alpha. Afterwards, beta is eotaxin, it is mediated by eosinophils, and MCP1 RANT is mediated by monocytes and macrophages. Okay, and what is C gamma? C gamma is lymphotactin, it is mediated by lymphocytes. Okay, and CX3C is fractalin, which is mediated by monocytes and T cells. So repeat with me. What is alpha? Alpha is mediated by neutrophils. CX X is like 8, so it is mediated by interleukin 8. Okay, what is CC beta? His eotaxin is eosinophils. Okay, and what is C gamma? Gamma is lymphotactin, which is mediated by lymphocytes. Okay, and CX3C is fractalin, which is mediated by monocytes and T cells. Okay. Coming to leukocyte recruitment. What is leukocyte recruitment? Okay, in leukocyte recruitment, we can remember with a mnemonic MRAD-M. Okay, what is MRAD-M? MRAD-M is nothing but margination, rolling, adhesion, diapedesis, migration. You know what is the mnemonic? It is M. Radiology madam, you can remember. I am radiology madam. Okay, that is how to remember the leukocyte, how the leukocytes all will come near the sites of inflammation. You have to remember with the mnemonic M rad M, that is margination, rolling, adhesion, diapedesis and migration. What is margination? Margination means leukocytes move to the periphery of the vessel on slowing of the blood, and when there is stasis, the leukocytes will move to the periphery of the vessel. Okay, what is rolling? Weak and transient attachments of leukocytes to the endothelium is called as rolling. In rolling, we'll see selectins bind to sialyl Lewis X on leukocytes. What is sialyl Lewis X? You can see in this picture. This is the selectins. Okay, this selectin, you can see one carrot-like structure. That is the sialyl Lewis X. Okay, so what happens in rolling? We can transient these all the leukocytes. The cell will attach to the endothelium. Selectins bind to sialyl Lewis X on the leukocytes. Carrot-like structure is is sialyl Lewis X. So when it has there are two types of selectins. There is E-selectin and P-selectin. E-selectin is present on the endothelium, whereas P-selectin is present on the platelets and endothelium. Okay, defect in this selectins is due to impaired fucosylation. So I told you selectins are these carrot-like structures. This in rolling, what happens is this transient attachment of leukocytes to the endothelium happens with the help of sialyl Lewis X. Okay, selectins bind to the sialyl Lewis X on the leukocytes. Selectins by the selectin is present on endothelium only. E-selectin is present in the endothelium. P-selectin is present in the platelets and endothelium. Okay, so defect in this SLEX is due to impaired fucosylation. Due to the impaired fucosylation, there is defect in this SLEX only. Okay, so it causes leukocyte adhesion deficiency type 2. So there are two types of LAD2. Okay, one is LAD type 1 where there is adhesion defect and integrin defect. Okay, how do you know if it is LAD type 1 defect? When a baby is born, there is a separation of cord is delayed. That is LAD type 1 defect. There we'll see adhesion defect and integrin defect. How can you remember this? LAD type 1, AD for adhesion defect, and type 1 means integrin defect. Okay, so but there is no pus formation, but there is delayed separation of umbilical cord. What is LAD type 2? LAD type 2, there is rolling is affected, means SLEX is only defect because of faulty glycosylation. Okay, it is associated with Bombay blood group. LAD type 2 is rolling defect. LAD type 1 is adhesion defect. Okay, now coming again, how to remember leukocyte recruitment? I told you I'm radiology madam. M rad M margination. First, in margination, what happens? Leukocytes move to the periphery of the vessel. When the blood slows down, leukocytes move to the periphery of the vessel. In rolling, what happens? Weak and transient attachment of the leukocytes to the endothelium. So they attach weakly with the help of what? Sialyl Lewis X on the leukocytes. This leukocyte will attach to the endothelium. On endothelium, there are two sets of endothelium. One is P-selectin, one is E-selectin. Okay, E-selectin is present in the endothelium. P-selectin is present on the platelets and endothelium. Okay, the defect in the SL Lewis X is due to impaired fucosylation. Okay, defect in this sialyl Lewis X, this carrot-like structure, is due to impaired leukocytation, and it causes LAD type 2 defect. Okay, I told you what is LAD type 1. Okay, that is adhesion defect. LAD type 2 is a rolling defect. It is associated with faulty fucosylation, and it is seen in Bombay blood group. Bombay blood group is a separate blood group. Okay, there will be defect in glucosylation. Now coming to M-RADM, rolling is over. Now coming to adhesion. In adhesion, what happens? Leukocytes firmly stick to the endothelium, mediated by integrins binding to ICAM1 on the endothelium. Okay, what are the integrins? Include LFA1, MAK1. Defect in the integrins causes leukocyte adhesion defect type 1. So what happens in adhesion? This leukocytes will firmly stick to the endothelium. They firmly stick to the endothelium, mediated by integrins binding to ICAM1 on endothelium. So integrins, you know the ICAM rhino mnemonic, okay, I came to see rhino. So this ICAM will bind. So this mediated by integrins bind to the ICAM1 on endothelium. What are the integrins? They are LFA1 and MAC1. Okay, LFA1 and MAC1 are the integrins. Okay, this defect in the integrins causes leukocyte adhesion deficiency type 1. Okay, and it is leading to delayed separation of cord, and there will be pus formation. Okay, what is diapedesis? Diapedesis in this MRAD-M. Margination is over, rolling is over, now addition is over. I told you about what happens in addition. In addition, look, LAD type 1 defect, if there is any defect in the integrins, causes type 1 defect. Okay. And in diapedesis, what happened? This leukocytes pass through the endothelium with the help of PCAM. Okay, diapedesis, leukocytes pass through the endothelium with the help of PCAM, that is CD31. In migration, what happens? Chemotaxis. Chemotaxis is caused by chemokines, I told you. Okay, major chemokines are LTB4, interleukin 8, C5a, 5-HETE and chalicorin. These are the leukotriene B4. Repeat with me: leukotriene B4, interleukin 8, C5a and C5-HETE. These are the major chemokines. Okay. So you can see the diagram here. First, in leukocytes will attach to the carotid substrate E-selectin. In this, if there is any fucosyl transferase defect, that will result in rolling effect, and it will call LAD type 2 SLEX defect. LAD type 2. LAD defect, that is SLEX type 2. In LAD type 1, what happens? There will be addition defect, integrin defect, and this is a delayed separation of cord. So, and there is a no pus formation. Okay. Coming to migration. Migration here is with the help of chemokines, interleukin C5, LTB4 and chalicorin. Okay. So CHC is with the help of chemokines, interleukin 8 and eotaxin. Okay. Coming to immunology, baby steps. Okay, what happens in immunology? There is major histocomparative complex. You know, in major histocomparative complex, there are two types of classes. One is class 1, class 2. Okay, what happens in class 1? Okay, in class 1, how is the structure of MHC? We have to know first. Okay, there are three alpha chains, and one is alpha 1, alpha 2. And this antigen binding groove is present between the alpha 1 and alpha 2. Okay, so this there is antigen binding groove, endogenous antigen binding groove is present here, and there are three, as you know, there are alpha 1, alpha 2, alpha 3. Class 1 has 3 chains. Okay, and there is one beta 2 microglobulin in the down. Okay, in MHC class 1. Okay, HLA, A, B, C all belong to MHC class 1. Okay, and in MHC class 2, it is due to DP, DQ, DR, all the 3 letter, and HLA on the chromosome 6. Okay, these are all MHC class 2. In MHC class 2, we have alpha 1, alpha 2, beta 1, beta 2, alpha 1, alpha 2, beta 1, beta 2. Okay, it is all in MHC class 1 is present on all the nucleated cells except sperms and platelets. Okay, is present in all the nucleated cells. We have MHC class 1. Okay, except the sperms and platelets. Okay, it is coded by the gene HLA BC. Okay, here protein binding cleft is made only of alpha chains. Okay, alpha 1 and alpha 2. It presents to the CD8 T cells. Rule of 8 is 1 into 8 is equals to 8. Endogenous antigens are recognized by virus infected cells and tumor cells. Okay. Here what happens? Here protein binding cleft is made of only alpha chains, alpha 1 and alpha 2. It presents to the CD8 cytotoxic T cells. Okay. Endogenous antigens are recognized such as virus infected cells and tumor cells. endogenous antigens are recognized such as virus infected cells and tumor cells. Whereas in HLA class 2, I told you MHC class 1 is present in all the nucleated cells except the sperms and RBC. Okay, in MHC class 1 what happens? There are 2 alpha chains, there are 3 alpha chains and 1 beta 2 microglobulin. Okay, except sperms and RBC, platelets, sperms and platelets, it is not present, because when the sperms enter the female cervix, there shouldn't be any immunity. The female cervix should absent the sperms, accept the sperms. That's why it is not present in sperms and okay, yeah. It is not present in sperms. Okay, so it's present in all the nucleated cells except sperms. Okay, it is also present in platelets. Okay, but protein binding cleft is made up of only alpha chains. It presents to this MHC class 1, though what will present to CD8 cell. So class 1 into 8, I told you the 1 into 8 is equals to 8. So MHC class 1 will bind to present to CD8 cells. What are the endogenous antigens here? These are the virus infected cells, tumor cells are the endogenous antigens. In MHC class 2, what happens? It is present in all the antigen presenting cells like B lymphocytes, dendritic cells, macrophages. Here MHC class 2 is present in all the antigen presenting cells. Okay, and hence antigen presenting cells have both MHC 1 as well as MHC 2. Antigen presenting cells they are also nucleated cells, so they will be having both MHC 1 as well as MHC 2. Okay, what are the especially the antigen presenting cells? There are B lymphocytes, dendritic cells, lymphocytes and macrophages. Okay, what is the protein? It is coated by the genes I told you already, HLA, DP, DQ, DR. What are the protein binding cleft made up of? It is made up of both alpha chains and well as beta chains. Protein binding cleft here is made up of alpha chains as well as beta chains. Why? Because in MHC class 1, alpha 1, alpha 2, alpha 3. Okay, so this protein and beta 2 microglobulin. So protein binding chains, it is made up of beta 2 microglobulin and alpha chain, one chain on one side, and alpha 2 and alpha 3 on another side. Whereas in MHC class 2, it is made up of alpha chain, alpha 1 and alpha 2, beta 1 and beta 2. Okay, in MHC class 2. So I told you already told you rule 8 table should be satisfied here. So here CD4 T cells will bind to MHC class 2. 2 into 4 is equals to 8. Protein binding cleft. And here exogenous antigens are recognized such as bacteria and toxins. Okay, this protein binding cleft is deep inside. So exogenous antigens are also recognized such as bacteria and toxins. Coming to antigen presenting cells. So what do the antigen presenting cells do? They take up the antigen and they present to the T cells. Okay, the antigen presenting cells can be remembered by B lymphocytes. Dendritic cells are the most important antigen presenting cells are the dendritic cells, macrophages and monocytes. Okay, the T cells then recognize by specific peptides on the surface of the antigens known as MHC molecules. Okay, what the T cells do, they recognize the specific peptides on the surface of antigens known as MHC molecule. Okay, in T cell activation signals, there is signal 1 and signal 2. Signal 1 is a T cell receptor. Okay, it recognizes antigen MHC. Okay, and see there are this now see what do antigen presenting cell, they take up the antigen present to C cells. Come see, this is antigen presenting cell, this is T cell. Okay, this antigen presenting cell, what it does, okay, it will represent it to the T cells. So in T cell activation signals, between this and this, there will be two signals, signal 1 and signal 2. Signal 1, what happens? The T cell receptor, this is T cell receptor, that is CD3 complex. Also it recognizes antigen MHC. Okay, signal 2, one signal is over. Now coming to signal 2. In signal 2, what happens? The B7 on the antigen presenting cell will attaches to the CD28 on the T cell. Okay, you can remember the mnemonic as the 4 7s are 28. 4 7s are 28 is that is B7 on antigen presenting cell will attaches to CD28 on the T cell. Okay. Signal 1 is over, that is CD3 complex. T cell just it will recognize. In second stage, signal 2, what happens? 4 7s are 28, that is 7 B7 on the antigen presenting cell will recognize CD28 on the T cell. this co-stimulatory action is required for the full activation of this signal. Okay, so what is this inhibit now to this co-stimulatory? The second one is the more major important one. This important one is inhibited by CTLA4 on the T cell. On the T cell there is a CTLA4. Okay, this is antigen presence on the this is T cell. On the T cell, this T cell activation is inhibited by CTLA4. Okay, so this CTLA4 is the signal which inhibits. Okay, in immune checkpoint inhibitors, we know some drugs which activate the CTLA4, which CTLA4 is inhibiting, so it will block the CTLA4 action and enhances the T cell activation. Okay, means that our body immunity only is overactivated here. Immune checkpoint inhibitors is a class of drugs which enhances our own body immunity. Okay, it is done in case of melanoma and renal cells cancer. CTLA4 inhibitor is used, means it doesn't allow it to inhibit the co-stimulatory action, so the T cell activity will increase here. Okay, it enhances T cell activation, which is done by ipilimumab. Okay, next, outcome of T cell activation. Now everything over, co-stimulatory action is over. First is this signal, first signal is over, second signal is over. Now T cells will get activated definitely. Okay, so in T cell activation, now we know the CD8 T cells which is present in MHC class 1, CD2 4 cells which is in MHC class 2. The CD8 cells kill the infected cells using perforins and granizymes. And what does CD4 helper cells do? CD4 helper cells will release TH1, TH2 and TH17. Okay, there are three types. The CD4 T helper cells are 3 types. One is TH1, second is TH2, and third is TH17. Okay, TH1 causes cell mediated immunity and granuloma formation, whereas TH2 causes humoral immunity, whereas TH17 is neutrophil recruitment. So TH1 releases interleukin 12 and interferon gamma. Okay, it produces interferon gamma and interleukin 12. Okay, it recruits the macrophages and enhances the killing of intracellular pathogens. Okay, TH1 will recruit the macrophages and enhances the killing of intracellular pathogens. It is involved in autoimmunity and chronic inflammation. Okay, it is a type 4 hypersensitivity reaction. Okay, IgG class switching in B cell inhibits the TH2. Okay, so what happens in TH1? There is granuloma formation. Okay, interleukin because of interleukin 12 and interferon gamma, so it enhances killing of intracellular pathogens. This is involved in the autoimmunity and chronic inflammation. It is a type 4 hypersensitivity reaction. Okay, in TH2 what happens? It is a humoral immunity. It activates B cells and antibodies. It produces interleukin 4, 5 and 13. Okay, interleukin 5 recruits eosinophils. Interleukin 4 is IgE class switching. It is involved in helminthic infections, allergic conditions. It is a type 1 hypersensitivity reaction. Interleukin 4 is a type 1 hypersensitivity reaction. Extra points, it also produces interleukin 10. It is anti-inflammatory. TGF beta plus interleukin 1 and interleukin 6. Okay, TH17. So TH7 is neutrophil recruitment. It produces interleukin 17. Okay. So coming to B cell activation, signal 1. B cells engulf the antigen and present to helper cells via MHC class 2. T cell recognizes antigen MHC. Whereas in signal 2 what happens? Now T cell immunity is over. I told you TH1, TH2, TH17. TH1 is cell mediated immunity. It releases helps in the granuloma formation. Okay, interleukin 12, and TH2 is responsible for humoral immunity. It releases interleukin 4 and interleukin 2. Okay, interleukin 4 and interleukin 2. Interleukin 4 is responsible for the IgE class switching, whereas interleukin 5 is responsible for the eosinophils recruitment. Okay, so interleukin 4 is also involved in helminthic infections and allergic conditions. TH17 is for neutrophil recruitment. It produces interleukin 17. Okay, TH17 is responsible for neutrophil recruitment. It produces interleukin 17. interleukin 17, that is TGF beta, interleukin 6, and it produces interleukin TGF beta, interleukin 1, and interleukin 6. Okay, that is over. Okay, next is how B cells will get activated. Here also there is two signals. One is signal 1 is B cells engulf the antigen and present to the helper T cells via MHC class 2. B cells will first engulf the antigen and present to the helper T cells via MHC class 2. Signal 2, here also your co-stimulatory signal is there, that is CD40 to 40 ligand interaction. Okay, this interaction leads to B cell activation. Okay, now coming to B cell here, first this is activated CD4 T cell. Okay, activated CD4 helper T cell will come with the T cell receptor will bind to MHC class to present on the B cells. So what happens is, now in the B cell it is like a bakery. Okay, in bakery you can make customized cakes, or you can go and buy ready-made cakes. Now what are the ready-made cakes here? IgM, IgD. Okay, it is present on the surface only, IgM, IgD. Okay, whereas to make the customized cakes, okay, the other IgG, AE, you need something called as chef to make customized cakes. So that chef here is nothing but CD40 to CD40 ligand interaction. Okay, the second signal which I told you, co-stimulatory signal. Okay, so this co-stimulatory signal will leads to this activation of IgG, IgA, IgE. B and D are present on the surface itself. Okay, so this when the CD40 to 40 ligand interaction is inhibited, this interaction leads to B cell activation. B cell initially produces only IgM greater than IgD. Class switching, IgJAE is produced by switching from IgM. Okay, switching from IgM. Affinity maturation and memory B cells formation is seen here. This IgGA is produced by switching from IgM, affinity maturation and memory B cells formation.